So again, in today's lecture, we are going to give a detail of the rest of the amino acids biosynthesis. So let's start from here that methionine, threonine, lysine, isoleucine, valine, and leucine are classified as essential amino acids, meaning that the human body cannot produce them and must obtain them from dietary sources. The biosynthetic pathways for these amino acids are intricate and interconnected, as you can see in the whole figure. Notably, the pathway may exhibit a significant variations among bacteria, fungi, and plants. The bacterial pathways are specifically illustrated in figure, highlighting the diversity in the synthesis of these essential amino acids across different organisms. The aspartate serve as the precursor for the synthesis of the methionine, threonine, and lysine. The critical branch points in these pathways occur at aspartate semialdehyde and intermediate present in all three processes, and at homoserine, a precursor for threonine and methionine. Additionally, threonine acts as a precursor for isoleucine. The pathways for valine and isoleucine share four enzymes. The synthesis of valine and isoleucine from pyruvate involves the condensation of two carbons of pyruvate in the form of hydroxyethylthiamine pyrophosphate with either another molecule of pyruvate leading to valine or with alpha ketobutyrate, which has been resulting in isoleucine. The alpha ketobutyrate necessary for isoleucine production is derived from threonine through a reaction requiring pyridoxal phosphate. Notably, an intermediate in the valine pathway, that is alpha ketoisovalerate, served as the starting point for a four-step branch pathway leading to leucine, as you can see the step 22 to 25 of the figure. This intricate network of pathways demonstrate the interconnected nature of amino acid biosynthesis and the shared intermediates in the synthesis of these essential amino acids. The chorismate is the key intermediate in the synthesis of aromatic amino acid such as phenylalanine, tyrosine, tryptophan. So now we will discuss their biosynthesis in detail. The formation of aromatic rings in tryptophan, phenylalanine and tyrosine occurring across bacteria fungi and plants, constitutes the primary biological pathway despite the inherent stability of the benzene ring. This pathway involves a branched process proceeding through the closure of a ring derived from an aliphatic precursor followed by the stepwise addition of double bond. In the initial 4 steps of this pathway, shikimate is produced, which is a 7-carbon molecule obtained from the combination of erythrose 4-phosphate and phosphoenolpyruvate, as you can see in this figure. The subsequent 3 steps involve the conversion of shikimate into chorismate, incorporating an additional 3 carbons from another molecule of phosphoenolpyruvate. Chorismate serve as the first branch point in the pathway, with one branch leading to tryptophan and the other to phenylalanine and tyrosine. This elaborate process underscores the complexity of aromatic ring formation and the strategic utilization of precursor molecules in the biosynthesis of essential aromatic amino acids. In the pathway leading to the synthesis of tryptophan, the process initiates with the conversion of chorismate to anthranilate. A key contributor to this transformation is glutamine, which donates the nitrogen required for the subsequent formation of the indole ring. Anthranilate, the product of this conversion, then undergoes a crucial step by condensing with 5-phosphoribosyl 1-pyrophosphate. This condensation sets the stage for the formation of the indole ring in tryptophan. The indole ring in tryptophan is a composite structure derived from the ring carbons and amino group of anthranilate. So, additionally, 2 carbons crucial for this process are sourced from PRPP. The final and decisive reaction in this sequence is orchestrated by the enzyme tryptophan synthase. The tryptophan synthase exhibits a complex alpha 2 beta 2 subunit structure. Interestingly, this enzyme can undergo dissociation into 2 alpha subunits and beta 2 unit. Each of these units plays a distinct role in catalyzing various parts of the overall reaction process, adding a layer of intricacy to the synthesis of tryptophan. The modularity of tryptophan's synthase allows for efficient and coordinated control over the multiple steps, as you can see in reaction, which is involved in the biosynthesis of these essential amino acids. In the second phase of the reaction, as illustrated in the figure, the participation of pyridoxal phosphate becomes essential. Notably, the enzyme retains the indole formed in the initial part and channels it through a passage from the alpha subunit active site to one of the beta subunit active sites. Within this beta subunit site, indole undergoes a condensation process with the Schiff base intermediate derived from serine and pyridoxal phosphate. This type of intermediate channeling appears to be a characteristic feature extending throughout the entire pathway from chorismate to tryptophan. Interestingly, the arrangement of enzyme active sites responsible for various steps in the tryptophan pathway varies among different species. In some fungi and bacteria, a single polypeptide hosts active sites for different steps, while in others, these active sites are distributed among separate proteins. Furthermore, the activity of specific enzymes within this pathway is contingent on non-covalent association with other enzymes involved. These observations collectively suggest that in both bacteria and eukaryotes, all enzymes participating in the pathway form part of a larger multi-enzyme complex. Despite challenges in preserving these complexes intact during isolation using traditional biochemical methods, accumulating evidence supports the existence of such multi-enzyme complexes in various metabolic pathways, including the one leading to tryptophan synthesis. In plants and bacteria, the synthesis of phenylalanine and tyrosine is achieved through a pathway considerably simpler than the intricate tryptophan pathway. These pathways share a common intermediate known as a prephenate. As illustrated, the conclusive steps in both cases involve the transamination with glutamate molecule. So as you can see that histidine biosynthesis need the ribose 5-phosphate, which is precursor of purine biosynthesis. Moreover, the pathway for histidine biosynthesis has been completely worked out in plants. 8 enzymes are involved in the biosynthesis of histidine. Complete pathway for histidine biosynthesis occurs in the plastids. The pathway to histidine in both plants and animals, that is bacteria, The pathway to histidine in both plants and bacteria diverges significantly from other amino acid biosynthetic routes. Histidine synthesis involves the contribution of three precursors as depicted in the figure. PRPP contribute 5 carbons. The purine ring of ATP contributes a nitrogen and a carbon, and glutamine provides the second ring, nitrogen. Key steps in this pathway include the condensation of ATP and PRPP, where N1 of the purine ring links to the activated carbon 1 of the ribose of PRPP, as you can see in the step 1 of the figure. Subsequently, there is a purine ring opening resulting in the linkage of N1 and C2 of adenine to the ribose as illustrated in the figure. The formation of the imidazole ring follows a reaction facilitated by the donation of a nitrogen from glutamine, as you can see in the step 5 of the figure here. One notable aspect of this pathway is the utilization of ATP as a metabolite rather than a high energy cofactor, a departure from typical metabolic processes. However, this unconventional choice is not wasteful as it aligns seamlessly with the purine biosynthetic pathway. After the transfer of N1 and C2, the remaining portion of ATP is released as 5-amino imidazole 4-carboxamide ribonucleotide. In short, it is called as AICAR, which serves as an intermediate in purine biosynthesis. Importantly, AICR is rapidly recycled back to the ATP, highlighting the efficiency and interconnected nature of these biosynthetic pathways. So, in conclusion, we can say that amino acids are a vital molecule with diverse functions in biological system. Understanding their classification, properties, biosynthetic pathway, and their function is essential for comprehending the intricate processes that govern cellular function and overall health. Thank you very much. This is the end of the today's lecture.